morning, students. Last class, we have discussed the topic about structure of anther and uh, structure of anther. There you have seen that in a structure of anther, there are four pollen sacs present in two lobes of anther, and those four pollen sacs, as they are present in one anther, so it is called as a quadrisporangiate anther. So this structure only, last class we have seen this structure. This is just repetition of this class, the previous class. So today we are going to read about microsporogenesis. That's the formation of microspores. That means it is help in the formation of pollen grain, and pollen grain contains male gamete in a flower. So as I have said last class only, these are the two lobes of anther. This is one lobe and these are two lobes. Let's start with this diagram which you have studied last class. This is your one microsporangium which contains these dense cells which we call it as a sporogenous cells. And last class only I told you this gap between from the outer to the inner, that is a wall of the microsporangium, that is the anther wall. What are the four layers of anther wall? Number one, I said the first layer is epidermis, after that endothecium, then 3 to 4 layer of middle layers, and the fourth layer is a tapetum. Tapetum plays an important role in the formation of pollen grain from these sporogenous cells. Later on, you'll come to know how it is playing that important role. Now these sporogenous cells, if I say this is stage 1, these sporogenous cells are tightly packed inside this microsporangium. As anther grows, as anther matures, then what happens? This microsporangium also increase in size. Once microsporangium increases in size, the sporogenous cells which is present inside, by dissolution of the callus which is present in between, the cells become easily separated from each other and they are developing certain spaces in between. So in that case, we call it as a pollen sac. We will no longer call it as a microsporangium. So after the formation of pollen sac, the sporogenous cells undergo meiotic cell division. As it is a sexual reproduction, so definitely the first division will be meiotic cell division. Sporogenous cells, we call it as now microspore mother cells. What we call that is microspore mother cell. So this is your microspore mother cell. Microspore mother cells is diploid once it undergoes meiotic cell division. Now, after meiotic cell division, it forms into 4 tetrad. Here 4, you know that after meiotic cell division, 4 daughter cells are produced. So here also 4 daughter cells are produced, and those 4 daughter cells are called as microspore tetrad. Tetrad means 4. According to those tetrad, how they will arrange, there are 4 different types of arrangement we can see. One, this is called tetrahedral. This is isobilateral, one after another linear, and this is digastate. 60% or 70% of angiosperm have in common type of microspore that is called as tetrahedral. So tetrahedral arrangement is generally seen in angiosperm. All these tetrahedral, when it matures more and more, all these tetrahedral or microspore tetrad separate from each other and forms haploid spores, and that's what we call it as a microspores. Let's come back. So we have started from here. This is sporogenous cells. This become, we call it as a microspore mother cells. Next time it is called, this is called microspores mother cells. And then after that it is forming your tetrahedral microspore. After tetrahedral microspore is formed, that means they are still attached. Once they are separated and form 4 microspore. Now each microspore is coated with outer and inner coat, and each microscope we call it as a pollen grain. So again, so what is microsporogenesis? Microsporogenesis is the formation of pollen grain from microspore mother cell, or microspore formation taking place from microspore mother cell is called as microsporogenesis. That means conversion of microspore mother cells to microspore is called as microsporogenesis. This is very important definition. After each pollen grain is formed, pollen grain has been formed. Pollen grain has got 2 coats. This is the plasma membrane of this pollen grain. Over there, over this, there is this layer is called as intine, and over the intine is a continuous layer which surround this pollen grain. That is a nucleus which is present in a pollen grain. This is just a pollen grain has been formed. Over that, you can see there is a layer which is called as a exine is marked. Now regarding exine. Exine is made up of sporopollenin. Hope you remember when we had discussed the function of tapetum. That time I told you the tapetum function is to secrete sporopollenin or help in the formation of sporopollenin. So sporopollenin is the most resistance material which is present in living world. So that is why it is not very easy. We can degrade any pollen grain because of this sporopollenin. Now, sporopollenin is, exine is made up of sporopollenin, you can see in the diagram, and intine is made up of cellulose. Now, exine, you can see again in the next diagram, if you go with the next diagram, pollen grain undergoes, here we have seen this is what? This is single nucleus. It will undergo mitotic cell division, but it is haploid. It undergo mitotic cell division, and after mitotic cell division, one nucleus divides into two nuclei. When the cell divides, that is followed by the cytoplasm, this is a callus wall formation. When the cell divides, two cell you get easily visible in a one pollen grain. The larger cell is called as a vegetative cell, and the smaller cell is called as a generative cell. This callus wall formation becomes more pronounced, and at the end we will find there is a lens-shaped cells which is floating in the larger cytoplasm or in a larger cell, and at that time we call it as a generative cell. Now regarding this sporopollenin, it is the most resistant material present in living world, but this structure is also not continuous structure over in time. There are certain pores or there are certain space which are left out to be coated with sporopollenin, and those spaces we call it as germ pore. Now what is the importance of this germ pore? If you think like this, if it had been like completely coated with sporopollenin, then what would happen? There will be no chance for growth of pollen tube, and if pollen tube would not be occur, then there will be no pollination. So that to prevent this thing, what has happened or what is modified, what has been modified here, the sporopollenin is not completely or not a continuous coating over the intine. Certain spaces are uncoated, and those spaces where it is not present, that forms a disc-like depression and is called as a germ pore. So germ pore helps in the growth of pollen tube. Regarding this generative cell, in 60% of angiosperm, we find the pollen grain is two cell stage. That means one is a tube cell, another one is a generative cell. But in 40% of case, we find this generative cell further divide into two male gametes. So in that case, we call it as a pollen grain is in a three-celled stage. Okay, so we have discussed about the microsporogenesis, where from it started from sporogenous cells present inside the microsporangium. The sporogenous cells turns into microspore mother cell. Microspore mother cells undergoes meiotic cell division. After meiotic cell division, it forms four tetrad, that is microspore tetrad. Each microspore tetrad is converted into pollen grain. Pollen grain undergo mitotic cell division and forms two-celled pollen grain. Most of the cases we get two-cell pollen grain, and some of the cases like 40% of angiosperm we get three-cell stage once this generative cell divides into two male gametes. Study of pollen grain is called as a palynology, and some people having an allergy with this pollen grain, that is what we call it as a hay fever. And let's discuss the certain pollen grains we can use as a preserve, or we can preserve it, or some pollen grains we can use as a food supplement. So that is called as like when the pollen grains are preserved as a in the form of a fossil, that's what we call it as a microfossils. The reason is very simple: because of the presence of this sporopollenin, which cannot be degraded by the action of any alkaline, any acidic environment, or any enzyme. So this is called as a pollen viability. That means a pollen can stay for a long time. That is what we call it as a pollen viability. Some pollens can stay for 2 or 3 days. Some pollens can stay for years after years. Okay, so this we have discussed about the microsporogenesis, and this part, that the development of pollen grain from microspore, tetrad, is called as a development of male gametophyte. Next class we'll start discussion with megasporogenesis, and then we'll proceed to pollination. Thank you, class.